Okay, chapter 21, we're only going to cover sections 2 and 3, but these sections are pretty intense and have quite a bit of information, a lot of math, a lot of equations. It covers the evolution of populations, and we're going to be doing some Hardy-Weinberg equations and things like that that you want to be sure that you're really familiar with. And I would highly encourage you within Pearson to click on the Study Area tab on the left-hand side of the page, and then it'll open up a page. You click on Study Access Area, then a drop down, you want to make sure that you choose chapter 21 in the drop down and hit go. And then you'll see that under concept 21.2 and concept 21.3, there's a video, there's an investigation, and there's a couple of activities. And if you're really having a hard time understanding these equations after this lecture, then I would highly encourage you to go and work on those so that you kind of get a better idea and get a better feel for the information in the chapter. So, just a little bit of an introduction. One common misconception that people have is that organisms evolve during their lifetimes, and they don't do that. Natural selection acts on individuals, but only populations evolve. Populations are the smallest unit that can evolve. So, if we look at an example with these large-beaked birds during a drought, are these finches. Drought happened, and the birds that had the larger beaks were more likely to crack the seeds because they were dried out and find food in order to survive. So before the drought, the birds had smaller beaks because that's all they needed. After the drought, more of the population had thicker, stronger, larger beaks because that's what was able to survive. So the population evolved, but the individual didn't. The individual didn't grow a bigger beak within its lifetime. Individuals that had larger beaks survived, producing more individuals, more babies, more offspring that had larger beaks, until eventually the population was made up of birds with big beaks. Microevolution is a change in allele frequency in a population over generations. So you remember an allele is a gene, right? And as that allele, the frequency or the percentage, the chance that those genes show up changes. Microevolution is a change in those gene frequencies in a population over generations. I would know that the three main mechanisms that cause allele frequency change are natural selection, genetic drift, and gene flow. And we're going to go in depth in these in just a minute. Only natural selection consistently causes adaptive evolution. So one of the big concepts that you want to make sure that you understand is this thing called the Hardy-Weinberg equation. And we use this to test whether a population is evolving or not. So genetic variation is required for a population to evolve, right? If we didn't have different genes in a population, everything would stay the same. We'd all look identical to one another. Animals would all look identical. But genetic variation doesn't guarantee that populations will evolve. One or more factors that cause evolution must be at work for the population to evolve. And remember, those might be natural select, genetic drift, gene flow, right? A population is a localized group of individuals that are capable of breeding with one another and producing fertile offspring. So for example, donkeys and horses can breed with one another and produce offspring, but they are mules, and those are usually infertile offspring. There's been like less than 5 cases where they actually reproduce. Populations are not always isolated from one another geographically, but individuals typically breed with other members of their own population. They stick with their population if they're a true species. So one example are these caribou in Alaska. There's 2 herds, a porcupine herd and this 40-mile herd. And although at one time they probably were an individual species, they evolved or they adapted to separate themselves in 2 different herds, and they breed within their herd only. And so now they've become subspecies of this one species of caribou. Even though their ranges overlap, they pretty much stick to their own subspecies. A gene pool consists of all the alleles for a loci in a population. So just to kind of review what those terms mean, an allele is, so you have your, let me get this thing up. So you have your entire gene here, right? And or your entire chromosome, which is made up of many genes. Those genes we're going to call alleles, right? So this is the specific gene for having short hair. This one is a specific gene for having white fur. And let's just call these hamsters, guinea pigs maybe. The locus is the location. Locus is the location where the allele is found. So a gene pool consists of all the alleles, all the genes for all the loci, all the spots in a population. An allele for a particular locus is fixed if all individuals in the population are homozygous for the same allele. Homozygous means that you have only recessive or only dominant traits. Remember, heterozygous means that you got one dominant trait and one recessive trait, maybe one from dad, one from mom, right? Homozygous means that both mom and dad gave you the same trait. So just looking at this again, a locus is the location of a gene or an allele, right? And this is so we have two chromosomes that are, two chromosomes that we get from mom and dad, two different alleles. Mom gives us one trait, dad gives us another trait. If they're both the same trait, it's homozygous. If dad gave you a different one than mom, it's considered heterozygous. The frequency of an allele in a population can be calculated. So we can figure out how often this trait shows up in a population. So we're considered diploid organisms because we get a trait from mom, a trait from dad. So the total number of alleles at a locus is the number of individuals times 2. Doesn't make sense right now, I'm sure. This will start to make sense. Just listen and kind of let it soak in. And as we keep going, this will make a little bit more sense to you. The total number of dominant alleles at a locus is 2 alleles for each homozygous dominant individual plus 1 allele for each heterozygous. So, and the same applies for recessive. So we have, for mom and dad, we've got, this is homozygous, this is homozygous. This is homozygous dominant, right? And then this is homozygous recessive. Dad gave us the dominant trait to be purple flower. Mom gave us the dominant trait to be a purple flower. Or dad gave us the recessive trait to be white, and so did mom. If mom gave you the recessive trait and dad gave you the dominant trait, you are heterozygous. Hetero means different. But what you look like, your phenotype, with how you look, how it's represented in the population, is of course that the dominant trait shows up. So you're going to be a purple flower. So if there are 2 alleles at a locus, moving over here, right? 2 alleles at a locus because we get one trait from mom, one chromosome from mom, one from dad. If there's 2 traits at a locus, there we are. There's 2 alleles at a locus, 2 genes at a locus. P and Q are used to represent their frequencies. So we use P and Q. Frequencies of the alleles population will add up to 1 because frequency, think of it as a percentage. So it's got to make a whole, which is 1. 100% of the population, 1. So P plus Q equals 1. I would just remember this. P plus Q equals 1. P being the dominant, Q probably being recessive, and it equals 1. And this is for only the alleles. This is only for your genotype, right? Or actually, I'm sorry, this is the phenotype. It's how you look. So if we're going to let's look at a wildflower population. And in this example, it's a little different than that purple flower. You've got what they call codominance. So this is dominant. Dominant trait, you're going to be red, or if you are dominant, homozygous dominant, you will be a red flower. If both mom and dad gave you the recessive gene, you will be a white flower. And this one's a little bit funny. Sometimes it happens where a little bit of the gene shows up, of the recessive gene, it kind of becomes a muted pink, a very light red flower. That means that mom gave you white and dad gave you red. Okay, so if you look at these flowers, where they're incompletely dominant for color, right? So if you get any of the recessive gene, if you get a recessive gene and a dominant gene, you're going to be pink. So let's say we have a population of, I think this adds up to 500 flowers. 320 in this field are red. 160 of them look pink, and so we know that they are heterozygous, and 20 of them are white, which means that they are homozygous recessive. So let's calculate the number of copies of each allele. P plus Q equals 1, right? Okay, so CR, color red, is the dominant trait. Well, we've got 320 in the population, and if they were red, they had to have both of the alleles that they carry, both of the genes from mom and from dad are red. So 320 times 2, because we got it from both mom and dad, plus 160, because we got one gene in this population of 160 pink flowers from mom or from dad. So 320 times 2, because they're both of the genes are dominant, and 160 is only one is that dominant trait. Add that all up, it turns out to be 100. I'm sorry, 800. Looking at the white, we have 20 white flowers. Both mom and dad gave us white, so it's 20 times 2 plus 160, because now we've got, right, 160 pink flowers, which means that we have 160 of the white gene. Add that all up, now you have 200. So 200 alleles in the population are white, 800 alleles are red. So to calculate the frequency of each allele, the percentage of each allele, well, 800 and 200 equal 1,000, right? So we've got 80% are red, 20% are white. The sum of the alleles is always 1. So it's 0.8 plus 0.2 equals 1. That equation is called the Hardy-Weinberg equation. And it describes the genetic makeup expected for a population that's not evolving. So as long as it's not evolving, that equation should be at 1. Equaling 1 means that it's not evolving. Genes are not changing. If the data observed for the population differs from the expected values, then that tells us that the population is probably evolving. So that's why this equation is important, because it tells us if things are happening or not. A population in which mating is random, so random mating, it's not chosen or artificial, and none of the mechanisms of evolution are acting, is in Hardy-Weinberg equilibrium. I would know what Hardy-Weinberg equilibrium means. Allele and genotype frequencies remain constant from generation to generation for a population in Hardy-Weinberg equilibrium. And Mendelian inheritance preserves genetic variation in a population. Hardy-Weinberg equilibrium describes the constant frequencies of alleles in such a gene pool. For example, the same population of 500 wildflowers and 1,000 alleles where, so that's what we figured out, right? So we had 500 wildflowers, but each wildflower has 2 genes, right? 2 alleles, so 500 times 2 is 1,000. P was the frequency of the dominant trait, and that was 0.8. And Q is the frequency of the recessive trait, and that was 0.2. So let's look at it a little bit closer in this figure. So we know we've already figured out P and Q, right? So these are all the alleles in this population, right? We're just going to put them in a bucket and pretend like this is it. So we can look and we can see that from this population here, what we should expect to come out is that it's an 80% chance that your mom will give you the red allele and a 20% chance that you'll get the white allele. Same thing with dad, identical, 80-20 chance. If you find that over time, more than 20% of that population is white, then probably evolution is occurring. Oops. Okay, the frequency of genotypes can be calculated using the rule of multiplication. So this P plus Q equals 1, that's telling you that's telling you your dominant and your recessive only. Let's say that we want to know, though, what is the frequency of the heterozygous allele? Well, there's another equation, P squared plus, because you're getting 2, right? Plus 2PQ plus Q squared equals 1. And that's the frequency of the genotypes. And I really hate this thing. It's the frequency of genotypes can be confirmed using the Punnett squared. I'll let y'all look at that a little bit closer because it's very, very confusing, and I think I would confuse you more if I went through this very much. So I'd rather do some practice equations with you instead. So if P and Q represent the relative frequencies of the only 2 possible alleles in a population at a particular locus, whatever that means, then P squared plus 2PQ plus Q squared equals 1. So P squared and Q squared represent the frequencies of homozygous genotypes, right? And 2PQ is the frequency of heterozygous genotypes. So if we are, say, given the heterozygous frequencies, we can figure out probably P and Q, or vice versa. If you're given one, you should be able to figure out the others. Okay, so P squared, if we look at our wildflowers again, P squared is what we expect to see in the dominant, homozygous dominant population. 2PQ is what we expect to see for our heterozygous guys, our pink flowers. And Q squared is our expected frequency of the white flowers, the homozygous recessive. The Hardy-Weinberg equation describes a hypothetical population that is not evolving. Let's make sure you understand that this is if it's in equilibrium, it's not evolving, right? In real populations, though, allele and genotype frequencies will be changing over time. 5 conditions for a population to be in Hardy-Weinberg equilibrium are rarely met in nature. So there's 5 conditions that have to be met in order for equilibrium to be occurring. And I would know these 5. 1, you're not having mutations. Even within a human population, you have mutations. 2, random mating. I guess in a human population, you can consider the mating random. 3, no natural selection is occurring. So in a human population, we may not necessarily see natural selection, but definitely in animal populations where you have environmental conditions and other things, natural selection is more obvious, I should say. Extremely large population size, yes, we see that in human populations, but if you look at maybe small tribes that breed within one another in smaller parts of the world where they're isolated or whatnot, that's not necessarily occurring. And no gene flow. Well, we definitely have gene flow within the human population, right? We have people coming from other countries that might have different genetic traits than we do in the United States or even in Texas. And so you have gene flow coming in, and that's why we start to see changes in hair color and eye color and height, skin color, and it goes on and on and on. Why we're a very diverse population now than maybe we were when it was a lot harder to come from other parts of the world or get to other parts of the world. Natural populations can evolve at some loci while being in Hardy-Weinberg at other loci. I'm not going to talk about that too much. So let's apply Hardy-Weinberg equation. So there's this disease or syndrome, I guess. It's a medical condition called PKU. And PKU is something, if you have a baby, you're tested for PKU within the first days of the baby's life. And PKU is a mutation of the genes. It's extremely low mutation. It doesn't happen very often. And what happens is the body can't process amino acids properly. They start to build up. And so it's not curable, but it's treatable, or you just have to be really careful with your diet. Can't eat certain things so that you don't have this buildup that occurs. So the PKU gene mutation is extremely low. It doesn't happen that often. Mating selection is random with respect to whether or not an individual is a carrier of PKU, and that allows it to stay low. Natural selection can only act on rare homozygous individuals. So you have, because it's recessive, rarely will you be recessive for it, homozygous recessive. And if you don't follow the dietary restrictions, natural selection occurs because you die off. So our population size is large because this occurs in humans, and migration really has no effect because in other parts of the world, every other population has very similar allele frequencies as well. The occurrence of PKU is so rare, it's only 1 in 10,000 births. So let's try to figure out what Q squared is based on that. If it's 1 in 10,000 births, then we know that it's recessive. So let's see, let's call it, let's just say, we'll say A is dominant and little a is recessive. So to have it, you must have AA, right? Or we could do Q. You are homozygous recessive. So 1 in 10,000 births are you QQ, Q squared. Let's divide 1 by 10,000 to get the percentage. Well, that's 0.0001. We know that Q squared is our phenotype, right? That's how we look. But it's also being recessive, homozygous recessive. So Q squared is 0.0001. It's a 0.0001 chance, 1 in 10,000 births, that you will look like, that your gene will be homozygous recessive. So what's our allele frequency? What is the frequency that we only have Q in the population? That's all we care about, is how often we have Q. Well, we know Q squared. We want to know Q. Q squared is 0.0001. Take the square root of 0.0001 and you get 0.01. Q equals 0.01. Now, y'all might look at that. That might actually supposed to be 0.000, but hopefully you understand what we're talking about here. So now we have Q. Well, we can figure out P if we have Q, right? We know that so this is what the book gives as an example, but let's break it down and make it make sense. We know that P plus Q equals 1. We know from what we did over here that Q equals 0.01. So if P plus Q equals 1, P plus 0.01 equals 1. Let's do our simple algebra. P equals 1 minus minus 0.01. Well, 1 minus 0.01 is 0.99 and Q is 0.01. So to express that dominant gene in the population, it's almost 100. It's a 99% frequency. To express that recessive gene, it's a 0.01 frequency. And as long as that isn't changing, then we're in Hardy-Weinberg equilibrium. Well, now let's figure out the frequency of the carriers. Carriers would be would be P and Q, right? You would be heterozygous. You are carrying it and you could pass it off to your offspring. And it would be rare indeed, right, if two heterozygous individuals had a child and both gave them that recessive gene. And when that happens, though, it does happen, but it's a very small percentage of the population. So if we know that P equals 0.99 and Q equals 0.01, we know that other equation of P squared plus 2PQ plus Q squared equals 1, right? Well, we just plug it in. So, well, we want to know what a frequency of carriers is. 2PQ. 2 times 0.99 times 0.01 gives us about 2%. So approximately 2% of the population is heterozygous. I know this is confusing. That's why I really encourage you to do the practices and the extra study material so that it just becomes really, really simple. This is one of those things where practice is going to make you understand it better. Okay, concept 21.3 is about natural selection, genetic drift, and gene flow and how it alters allele frequencies in a population, how it changes the way things look in a population. And so this is less equations and more vocabulary and content material. 3 major factors that alter allele frequencies and bring about most evolutionary change are natural selection, genetic drift, and gene flow. So natural selection is success in reproduction results in certain alleles being passed on to the next generation. There's something that is good about a certain gene that mother and father pass on to the next offspring, allows them to survive, and it goes and goes and goes, right? So one of the best examples is when we use DDT to try to control insect populations, and we used it in agriculture. DDT was a very powerful insecticide. It knocked out insects really wonderfully, but we overused it. We relied on it too much. And we also used it for mosquito control too. So what happened was we used DDT, and let's say that this is a small portion of the population. Let's say that each mosquito represents 10,000 mosquitoes. Well, you end up with, let's say, 2% of the population survives. For some reason, they have a certain gene that allows them to survive. You spray DDT, these guys survive, and they make more offspring that carry the gene. And this is very simplified because there are many, many, many steps in between here. These two would pass, for whatever reason. Now they passed the gene on, they survived. The next time you sprayed, instead of killing 80% of the population, you only kill 40%. Next time it's only 20%. Pretty soon you're not killing anybody because you have selected for those who don't die from DDT. You've killed off everyone else that does, and now the only ones surviving are those that are resistant. So that's an example, a great example of natural selection. Genetic drift describes how allele frequencies fluctuate unpredictably from one generation to the next. The smaller the sample, the more likely that chance will cause some deviation from your predicted result, because you have less of a gene pool to choose from, right? You have less genetic variation. Genetic drift tends to reduce genetic variation through losses of alleles, especially in small populations. So in this example of these wildflowers, the plants that are highlighted are the only ones, for whatever reason, that leave offspring. So over time, fewer and fewer leave offspring, and then pretty soon, because you only had a few that left offspring, they're only passing on their genes. And so as subsequent generations occur, you're going to eventually knock out a whole allele sometimes. So same pictures. The founder effect occurs when a few individuals become isolated from a larger population. You've got a body of water, or you have a peninsula, a little island breaks off of the peninsula, or you have a river that all of a sudden starts flowing through, a creek or something that flows through and isolates a population. And so a few individuals are stuck on this little island. from the larger population, and so they have fewer genes, and they might eventually become a different species. Allele frequencies in the small founder population can be different from those in the larger parent population just due to chance, because randomly you ended up with different genes or specific genes from the parent population. The bottlenecked effect can result from a drastic reduction in population size due to a sudden environmental change. By chance, the resulting gene pool may no longer be reflective of the original population's gene pool. If the population remains small, it can be further affected by genetic drift. So the bottleneck effect would be where you have an original population, something drastic happens like a huge drought, or a huge drought that makes it so that there's no food for you to consume anymore. So only a few of the population survive. And it's just random who survives. Or, okay, let's give a better example. A hurricane comes through. You have your original population, a hurricane comes through, it knocks out most of your population randomly. It has nothing to do with survival, fitness, or anything else. It just happens that earthquake happens, hurricane happens, and bam, a bunch of your population dies. What survives, the surviving population, looks nothing like the original population. And so when these guys continue to produce, they will look very different. This population here, their babies, will look very different from what they did originally before the major natural disaster happened. Understanding the bottleneck effect can also increase our understanding of how human activity affects other species. So sometimes our activity can be a major natural disaster or unnatural disaster on another species. And one example is this little, the greater prairie chicken. So we reduced prairie habitat and caused the reduction in the population of these guys in Illinois. But the population stayed the same, and we didn't reduce the prairie in Kansas and Nebraska. And so this is a good example because you can compare what happened in Illinois, where we did damage, to what stayed the same in two other states. So in Illinois, the surviving birds had very low levels of genetic variation, and only 50% of their eggs hatched for some reason. Stress, maybe? Genetic drift during the bottleneck may have led to genetic loss of variation and increase the frequency of harmful alleles. So if we look at it, this was what they had. Well, we reduced all the prairie, and now they have a little bitty tiny population. So we had a major bottleneck where you've got only a few that survived. And what we see is that the population drastically went down by less than 50% of what it was, compared to Kansas, which has close to 1 million. Nebraska had no bottleneck. It has up to 200,000. We only have 1,025,000, drastically smaller than the other places. And what we see is that you have less eggs that hatch. The number of alleles per locus is smaller. Things are, 99% and 96% are hatching. Almost all of their offspring are hatching. Ours are getting much smaller. In 1993. So less than, actually, I'm sorry. So we had 25,000, now we have less than 50. 93% were hatching before, less than 50% are hatching now. So when you reduce population size, humans had an impact on that. We reduce the fitness of this little bird. Researchers use DNA from some of the museum specimens to compare genetic variations in the population before and after the bottleneck, and what we saw was that there was a loss of alleles at several loci, so a loss of genes at several locations. And when researchers started to introduce the chickens from populations in the other states, they introduced those alleles again, and we increased the egg hatch rate to 90%. So by getting rid of certain genes, we decrease the ability of these guys to reproduce. By bringing those genes back in, we were able to increase their ability to reproduce. So if we summarize this and look at it, genetic drift is very significant in small populations. I would it shows up much greater when you have small populations because you just don't have the genetic variability. Genetic drift can cause allele frequencies to change at random. Genetic drift can lead to the loss of genetic variations within populations, and it can cause harmful alleles to become fixed, ultimately reducing the population more. Gene flow consists of movement of alleles among populations. So we did gene flow, right, when they introduced other populations of chickens, prairie chickens from other states. Gene flow is the movement of alleles among populations. Alleles can be transferred through the movement of fertile individuals or gametes like pollen, right? And gene flow tends to reduce genetic variation among populations over time because it increases, right? Gene flow can decrease the fitness of a population. One example are these little Dutch birds, the great tits, and they immigrated from the mainland. So there's different populations of these guys. There's a mainland population, and then there is an eastern population. And when the mainland population started moving over into this little island population, it decreased their fitness because on the mainland population, there were certain things that allowed them to survive, right? So they looked a little bit different. Whatever genes they had allowed them to survive better. Well, those that were in the island populations also had their own genes. When you brought them over and introduced different genes on that island population, you introduced genes that maybe weren't so beneficial for them. And so the populations changed and they became less, their survival rate got much reduced over time. So females that were born in the central population that moved over to the islands didn't survive very well, right? Pretty low. But females that were born there in their eastern population on that island did survive very well because they got genes from mom and dad that allowed mom and dad to survive that passed on to their offspring. Gene flow can also increase the fitness of a population. For example, when you look at resistance to insecticides, so using mosquitoes as an example again, when we target mosquitoes that carry West Nile and other diseases and you spray for them, while this is a bad example for humans, this is a good example for the mosquitoes where it has increased their fitness, right? So we're spraying for them, but we are choosing those that are resistant to the insecticide, and they're passing those genes on, just like with DDT, to other offspring so that they are also resistant. Gene flow is an important agent of evolutionary change in modern human populations, which is just kind of a summary of what gene flow is, I guess. So I would know what gene flow, genetic drift, bottleneck effect, all those terms mean. I would understand Hardy-Weinberg equation. I know we only really used one example, but if you were given the allelic frequency of a heterozygous individual, could you tell me what the homozygous dominant was? Or if I gave you homozygous dominant, could you tell me, what heterozygous was? Be able to know that p plus q equals 1, and then p squared plus 2pq plus q squared equals 1 also. Plug in those numbers and be able to work with it. Again, I would go into Pearson. When we can get into Pearson, click on study area, access the study area, choose chapter 21, and do those extra videos and investigations and activities. After you've listened to this lecture, if you haven't already done so, do the dynamic study module. Those are going to be helpful as well. Your homework is also tutorial, giving you some examples of Hardy-Weinberg. And then finally do the quiz. Don't do the quiz before you've done the other two things, because those dynamic study modules and homeworks are meant to help you along with those quizzes.